Now it is my pleasure to introduce our first presenter, Dr. Amanda Jones. Dr. Jones is a lead pharmacologist in the Division of Bioequivalence 1, Office of Bioequivalence, in the Office of Generic Drugs within CDER. She will be joined by Drs. Darby Kozak and Dr. Hee-Sun Chung. And now I will hand it over to Dr. Jones.
Thank you for that introduction, and thank you all for joining us for this SBIA webinar on the new draft pH adjuster guidance. I will begin the webinar by providing the objectives, regulatory background, and the aim and scope of the guidance. When I am finished, I will hand it over to my colleague, Dr. Darby Kozak, the Deputy Division Director for the Division of Therapeutic Performance 1 in FDA's Office of Generic Drugs. Dr. Kozak leads a group of interdisciplinary scientists on the development of new analytical methods and methodologies to evaluate the equivalence of complex generic drug substances and complex generic parenteral, ophthalmic, otic, and inhalation formulations. Prior to joining FDA, Dr. Kozak was Chief Scientist for Eyes on Science and Research Fellow at the Australian Institute for Bioengineering and Nanotechnology. Dr. Kozak has a Bachelor of Science in Chemical Engineering from the University of Washington in Seattle, Washington, and a PhD in Chemistry from the University of Bristol in the United Kingdom. Today, he will summarize the role of pH adjusters and discuss supportive information to justify a difference in pH adjuster. Dr. Kozak will then hand it over to our colleague, Dr. Hee-Sun Chung, a lead pharmacologist in the Division of Bioequivalence 1 in the Office of Bioequivalence in the Office of Generic Drugs. She has more than 11 years of experience with bioequivalence assessment of generic products and currently leads a team of pharmacologists. She participates in various multidisciplinary working groups addressing complex scientific and regulatory issues. Hee-Sun received her Bachelor of Science in Chemistry and Master of Science in Inorganic Chemistry from the University of Oklahoma and PhD in Pharmaceutical Sciences from the University of Michigan. Today she will walk us through some different examples of when a 314.99b waiver request may or may not be appropriate, as well as some of the procedural aspects of submitting such a waiver request. Specifically, the objectives of this webinar are to provide you with an overview of the new draft guidance, describe the rationale for the agency's recommendations requesting 314.99b waivers for qualitative and quantitative differences in pH adjusters between the proposed and reference-listed drug formulations, discuss examples of data which may be considered in support of such waiver requests, and discuss timelines and the process for submission and mechanisms to receive feedback from the agency. I will start off by discussing the regulatory background and aim and scope of the guidance. Section 505J4H of the FD&C Act prohibits approval of an ANDA either if the inactive ingredients are unsafe for use under the conditions prescribed, recommended, or suggested in the labeling proposed for the drug, or the type or quantity of inactive ingredients included or the manner in which the inactive ingredients are included is unsafe for use under such conditions. FDA has interpreted this to mean that it gives us the authority to deny approval of an application if there is a reasonable basis to conclude that one or more of the inactive ingredients of the proposed drug or its composition raises serious questions of safety or efficacy. This allows FDA to identify changes in inactive ingredients or composition that may adversely affect a drug product's safety or efficacy based on our experience with reviewing inactive ingredients and from what other information we have available that speaks to it. This is codified in our regulations at 314.127A8 Romanet 2A. Additionally, we further specify in our regulations at 314.94A 9 Romanet 3 and 4, which are the inactive ingredient requirements with respect to the formulation of a drug product, and 314.127A8 Romanet 2B and C, which are the corresponding approval provisions for an ANDA, that FDA would consider an inactive ingredient in or the composition of a generic drug product intended for parenteral, ophthalmic, or otic use to be unsafe unless it contains the same inactive ingredients, with certain listed exceptions, in the same concentration as the reference listed drug, known as the RLD. And it is important to note here that this is not the reference standard. So when we're talking about same inactive ingredients, that's referencing the qualitative sameness, or Q1, while the same concentration refers to quantitative sameness, or Q2. The aim and scope of putting out this guidance on considerations for waivers of differences in pH adjusters using 314.99b is intended to assist applicants that reference an RLD intended for parenteral, ophthalmic, or otic use, but are seeking approval of a drug that is Q1 or Q2 different from the RLD with respect to pH adjusters, to share the agency's scientific considerations for why certain Q1 or Q2 differences in pH adjusters may be acceptable in a specific ANDA, and enable flexibility in the inactive ingredient requirements around pH adjuster differences to help remove some of those barriers to the development and approvability of drugs intended for parenteral, ophthalmic, and otic use where scientifically appropriate using the existing 314.99b waiver provision. So when is submission of a 314.99b waiver request necessary for a particular ANDA? Let's first discuss scenarios of qualitative or Q1 differences. Let's say the RLD contains pH adjuster A, but this pH adjuster is omitted in the test product. Or the opposite, the RLD does not contain a pH adjuster, but the proposed test product contains pH adjuster A. Or we have a third scenario where the RLD contains pH adjuster A, and while the test product contains a pH adjuster, it is different than that in the RLD, which is referred to as pH adjuster B in this table. For all three of these scenarios, these are not considered Q1 the same, and hence submission of a 314.99b waiver request would be necessary. To the extent that a pH adjuster is used to convert the active pharmaceutical ingredient, or API, to form the API salt, for example, in situ conversion of the API to a salt form, or use of a solubilizing agent in a similar manner, the pH adjuster is not included in the Q1, Q2 assessment because it becomes part of the active ingredient. If excess amount of pH adjuster is used beyond that needed to convert the API, that excess amount would generally be considered an inactive ingredient subject to the Q1, Q2 assessment. And Darby will speak more to this during his portion of the presentation. Now, when is a 314.99b waiver request submission necessary for a particular ANDA with respect to a quantitative or Q2 difference? When a proposed drug product formulation contains more than a 5% difference in concentration of a pH adjuster compared to the RLD, a waiver request citing 314.99b for that difference in concentration is necessary. Let's look at some hypothetical scenarios. Anytime the RLD lists the pH adjuster as QS, and since QS really can be reflective of any amount, this means that any amount in the proposed test product, whether it's QS or fixed, would be considered Q2 the same. So for Q2 differences, the focus should really be around instances where the RLD contains a fixed amount of the pH adjuster. So when the RLD does contain a fixed amount, but the proposed test product contains a QS amount, that is not considered Q2 the same, since QS is interpreted to mean potentially any amount, and some of those quantities could fall outside of the 5% window compared to the fixed amount on the RLD. Additionally, if the RLD and the proposed test product contain a fixed amount, we would need to compare the amounts, and if that difference is more than 5%, you would need to submit a 314.99b waiver request. If the difference is within 5%, there is no need for submission of such waiver requests. So what does this guidance do? It describes the scientific principles regarding the role of pH adjusters in formulations that underlie FDA's current thinking on why certain differences in pH adjusters may be acceptable in a specific ANDA. It describes how FDA intends to evaluate a request for a waiver of inactive ingredient requirements for a Q1 or Q2 difference in pH adjuster, And this guidance also includes recommendations on the type of information that you should consider submitting with your waiver request to help FDA in our consideration, and the format and process for submitting such waiver requests in your submission. Importantly, the guidance makes it clear that waiver requests will be evaluated on a case-by-case basis to determine whether a particular difference in pH adjuster is scientifically acceptable and appropriate in a particular ANDA, and that evaluating a waiver request is a fact-specific assessment within the context of a specific ANDA and the specific 314.99B waiver requests being made. And that wraps up my part, and I now hand it over to Darby.
Thank you, Dr. Jones. I'm Darby Kozak, the Deputy Division Director of the Therapeutic Performance 1 in the Office of Research and Standards in the Office of Generic Drugs. Over the next few slides, I will discuss some of the fundamental chemistry behind the role of pH adjusters and how this information, along with other information, can help support a proposed difference in pH adjuster compared to the RLD product. In particular, I will discuss the scientific background and justification to support a pH adjuster waiver request. These can be loosely categorized as when a pH adjuster acts as alone, when a pH adjuster acts as part of a buffer, and when a pH adjuster acts as an in situ converter of another ingredient in the formulation to convert that ingredient from the acid-free base to the salt, or vice versa. The next session will give a few examples and general thinking of when a pH adjuster acts alone. As a reminder of the fundamental chemistry, a pH adjuster is commonly a strong acid or base that is used to change the pH of a formulation by changing the equilibrium concentration of hydronium ions. The amount of pH adjuster used is therefore on a quantity sufficient to achieve a desired pH. At the same time, the pH adjuster contributes a counterion to the formulation. Increasing the amount of an acidic pH adjuster increases the amount of both hydronium ions and counterions proportionally. In the example shown here, the amount of hydrochloric acid pH adjuster added to the formulation gives an equivalent amount of both hydronium and chloride ions. Therefore, when there are no other ingredients that can act as an acid or base, a measure of the hydronium ion concentration, that is the pH of the solution, is correlated to the amount of HCl added. Moreover, the role of pH adjuster is often to achieve a desired formulation pH. Most drug products therefore express the quantity of a pH adjuster to be used as QS. This means that the quantity added is as much or as little as necessary to achieve a specific pH range. In some instances, the pH adjuster may not be used at all, but a drug product manufacturer may elect to include one as part of their manufacturing control process. Therefore, the pH range of the drug product is the primary aim, and the amount of pH adjuster used to achieve the pH of the drug product is adjusted accordingly. Examples of possible quantitative Q1 differences in pH adjuster can include use of a different pH adjuster, such as potassium hydroxide rather than sodium hydroxide, omission of a pH adjuster in the test product, such as electing to not include the use of sodium hydroxide when the RLD has listed use of that sodium hydroxide for pH adjustment, and the addition of a new pH adjuster, where the generic may elect to include use of a pH adjuster not used in the RLD, such as including sodium hydroxide when the RLD has not listed that use for that ingredient. Possible quantitative or Q2 differences that may be applicable to the waiver request is when the RLD lists a fixed amount of pH adjuster to be used, and a generic applicant proposes to use an amount quantity sufficient or QS, or when the generic proposes to use a fixed amount that may be greater than 5% different than the concentration used by the RLD. A waiver request and associated information to justify a waiver request is specific to the proposed product and associated proposed difference in pH adjusters. However, it's generally recommended that an applicant identify the proposed Q1 or Q2 difference in pH adjusters compared to the RLD as part of the request to waive a difference in pH adjuster for a parental ophthalmic or otic product. It's understood that the level of detail that an applicant can provide in this instance may be limited to the public information, such as the approved drug product labeling, as well as the reverse engineering data an applicant may have for the RLD product. Regardless, it is helpful to outline the general understanding of what differences are being proposed when compared to the RLD. In addition, it is recommended that an applicant provide information on the role of the pH adjuster difference, as well as the rationale and justification the proposed difference in pH adjuster does not affect the safety and efficacy of the drug product. This may be similar to what may be needed to support a difference in exception excipients. For example, the information that may be needed to support a change in buffer, which is a permissible exception excipient difference under 314.94a9. Although not limited to the following information, it would generally be recommended that an applicant include information from the inactive ingredient database, that the proposed pH adjuster and the amount that is being proposed has been in a previously approved FDA product for the same route of administration, that the difference in pH adjuster does not change the physical chemical properties of the drug product, as well as any other information that may be relevant to support the proposed difference. In the next section, I'll discuss examples of when a pH adjuster is used as part of a buffer system. As a reminder, a buffer is an aqueous solution of either a weak acid and its conjugate base or a weak base and its conjugate acid. This buffer system is resistant to changes in pH upon addition of additional acidic or basic components. A buffer is an exception excipient under 314.94a9. However, pH adjuster ingredients used to tune the pH of the buffer are not considered to be an exception excipient. Therefore, waiver requests may be submitted for proposed differences in pH adjusters used with a buffer system. It's important to note that a difference in buffer can change both the counterion and the pH of the system, similar to what a pH adjuster does. So similar information and justification may be considered to support a difference in pH adjuster as is used to support a change in formulation buffer. However, when a pH adjuster is added to shift the equilibrium between the conjugate base pair, the pH adjuster becomes part of the buffer. An example of this is when sodium hydroxide is added to an acetic acid sodium acetate buffer system, as this addition converts some of the acetic acid to sodium acetate. Similarly, sodium hydroxide added to a sodium phosphate monobasic and dibasic phosphate buffer system converts some of the monobasic to dibasic sodium phosphate. No new counter ions are generated, only a change of the ratio of the buffer component concentrations. A quick review of the fundamental chemistry of buffers helps illustrate this. Using a more complex buffer as an example, phosphate buffer, when in solution, phosphate monobasic ions are in equilibrium with phosphate dibasic ions. The equilibrium can be written as a phosphate monobasic dissociates into a proton and a phosphate dibasic ion with an associated dissociation constant of K2. The ratio of the phosphate dibasic concentration to the monobasic concentration in the solution is equal to 10 to the power of the pH minus pK2. As the dissociation constant K2 is a fixed property of the buffer, the ratio of the di to monobasic is solely dependent on the pH of the system. What this means is that no matter what the starting amounts are for the dibasic and monobasic sodium phosphate, at a fixed pH, the molar ratio will be the same. Therefore, if the total phosphate amount to the generic formulation is the same to that of the RLD, the individual dibasic and monobasic phosphate amounts are the same when the pH of the generic and the RLD solution are the same. So in brief, we understand that there are multiple ways to make the same buffer, as once these ingredients are in solution at a specific pH, they're no longer the starting materials, and instead they're in the same equilibrium ratio. That is, the same ratio of mono and dibasic phosphate will be present in all of these instances if they're at the same pH. This is regardless if the initial ingredients added were phosphoric acid and sodium hydroxide, or a combination of mono and dibasic phosphate, or any combination of these starting ingredients. The important thing is the concentration of phosphate and pH of the solution. However, under the current 314.94a9 regulation, any differences in the pH adjuster would need to request a waiver under 314.99b to be eligible for ANDA submission. Those with differences only in the phosphate buffer ingredients would generally be eligible under the current 314.94a9 regulations and would not warrant a 314.99b waiver. But as illustrated by this slide, there are a number of potential Q1 and Q2 pH adjuster differences in the starting ingredients. Information and rationale, such as outlined in this slide and the previous slide, may be included to support that such differences do not change the safety or efficacy of the proposed product. Examples of possible qualitative Q1 differences in a pH adjuster include the omission of a pH adjuster in the test product, such as electing not to include the use of sodium hydroxide when the RLD has listed use of the sodium hydroxide, including a pH adjuster not in the RLD, but with the same counterion as the buffer, such as using sodium hydroxide in a sodium phosphate buffer when the RLD has not included the use of a sodium hydroxide for pH adjustment, and including a pH adjuster with a new counterion that's not used in the RLD, such as using potassium hydroxide rather than sodium hydroxide in this example. As mentioned previously, possible quantitative or Q2 differences that may be applicable to a waiver request is when the RLD lists a fixed amount of pH adjuster and the generic applicant proposes to use a QS amount or a fixed amount that is greater than plus or minus 5% different to the concentration used in the RLD product. The same fundamental principles and rationale can be applied to the other buffer systems. This includes the use or omission of sodium hydroxide in acetic acid sodium acetate buffer, or the use or omission of a sodium hydroxide in a citric acid sodium citrate buffer system. In both instances, the rationale support that the total buffer ion concentration and the pH of the system is the same between a proposed test product and the RLD can help support a waiver request for a proposed difference in the pH adjusters used. Information to support a waiver request of differences in pH adjusters used in a buffer formulation will be product and formulation specific. However, it is generally recommended that an applicant identify the proposed Q1 or Q2 difference in pH adjusters used compared to the RLD. This will get a general outline and understanding of what differences are being proposed and compared. It is understood that the level of detail that can be provided may be limited due to the public information and reverse engineering data that an applicant may have. In addition, it's recommended that an applicant provide information on the role and interaction of the pH adjuster with the buffer and any potential differences to the RLD, as well as the rationale and justification that the proposed differences does not affect the safety and efficacy of the drug product. This may include, but is not limited to, concentrations of the newly formed salt or acid-base species in the buffer versus the IID limits for those components of the same route of administration, the physical-chemical characterization of the proposed formulation. This can include things like the pH, buffer capacity, osmolality, viscosity, compared to the reference product, and any other information that may be beneficial to support the proposed difference in pH adjuster. A key aspect of the supporting information is to clearly lay out the rationale for why the proposed ingredient difference does not raise a safety or efficacy concern compared to the RLD formulation. In the next few slides, I'll discuss examples when a pH adjuster acts as an in situ converter to change the salt form of an ingredient other than the active pharmaceutical ingredient or a buffer. When used to adjust the pH of the formulation, a pH adjuster may also interact with other ingredients in the formulation to form a salt or the free acid or base of that ingredient. For example, a simple neutralization reaction as shown on this slide can occur where a basic and active ingredient is neutralized by the addition of hydrochloric acid pH adjuster. In the process of reducing the pH, the hydrochloric acid also changes some of the basic ingredient from the free base to the chloride salt. This represents the in situ conversion of that ingredient, which give rise to the same chemical composition as would be if you were to add the salt form to the formulation. A pH adjuster used as an in situ converter is similar to when a pH adjuster is used with a buffer. For a buffer, only a fraction of the buffer ingredient is converted. In contrast, when a pH adjuster is used as an in situ converter, it is generally expected that the pH adjuster is a strong acid or base and is used on an equal molar amount to the inactive ingredient being converted, such that the entire amount of the inactive ingredient is converted. As mentioned earlier, pH adjusters used to form the active ingredient is not considered as part of a Q1, Q2 assessment, as formulation assessments are for the inactive ingredients in a formulation. But it is recommended that applicants clearly identify if this is the case and provide supporting information that the equal mole amount is being used. It's important to note that excess amount of pH adjuster used beyond that used for the in situ conversion are generally considered to be an inactive ingredient that would be part of a Q1, Q2 assessment. Therefore, as discussed in more detail in the next slide, it's recommended that applicants clearly outline the role and amounts of pH adjusters that may be used for in situ conversion of another inactive versus those that may be used in excess to adjust the pH of the formulation. In the example on this slide, sodium hydroxide can be used in equal molar concentration to convert benzoic acid, a standard preservative, to the formulation to form sodium benzoate. Like with the previous examples of a proposed difference in pH adjuster, the information to support a waiver request will be product and formulation specific. Where possible, applicants should identify the proposed Q1 or Q2 difference in pH adjusters to the RLD, should supply information from the IID that the in situ formed ingredient and associated pH adjusters have been used in a previously approved product for the same route of administration at amounts equal or less than that being proposed. In addition, an applicant should include information on the comparative characterization of the proposed product's physical-chemical properties and compare that to the reference product, as well as include any other information that may be beneficial to support the proposed difference in pH adjuster. With that, I'll hand you off to my colleague, Dr. Hee-Sun Chung, who will talk more on the practical applications of this guidance.
Thank you, Dr. Kozak. I will start my portion of the presentation by talking about will deny and may deny situations. The agency will deny a waiver request when the difference in pH adjuster is not acceptable for an ANDA. For example, if the difference in pH adjuster causes the final product to contain a different form of active ingredient than the RLD, or a novel inactive ingredient that has not been used in an FDA-approved drug product, the safety of which cannot be established without clinical testing. The agency may deny a waiver request if the difference in pH adjuster impacts physical or chemical properties critical to the performance of the drug product, or where those property changes raise potential safety concerns. For example, ANDA uses a different pH adjuster compared to the RLD, and that difference gives rise to either a new counter-ion species not present in RLD, or a different concentration of counter-ion species than the RLD. A change in counter-ion concentration or species may impact the physical-chemical properties of complex formulations, which may alter performance of the drug product in ways that may not be appropriate for approval in an ANDA. For example, the final pH of the test product is very different from the pH of the RLD product. Now let's take a look at some hypothetical examples. I would like to note that the hypothetical examples provided in next couple of slides are provided to highlight some of the agency's considerations for evaluating a 31499B waiver request during the scientific assessment of the ANDA. Examples are intended to illustrate instances when a 31499B waiver may be acceptable in an ANDA but do not constitute grant scenarios. As Dr. Jones mentioned, a decision to grant a waiver request is based on scientific evaluation of the specific facts of an individual ANDA, including the supporting documentation and justification submitted for a specific waiver request. I would also like to note that in the examples provided, the formulation information includes just sufficient details to illustrate the example. The first example is for an ophthalmic solution. The test and reference products are Q1 the same, as they both contain sodium hydroxide as pH adjuster. However, because the difference in the amount between test and reference products is more than 5%,
they are not Q2 the same with respect to sodium hydroxide. It may be acceptable to waive the inactive ingredient requirement if scientifically justified. The possible supporting data for the 31499B waiver request is comparable physical-chemical data between the test and the reference standard products. The second example is for an injection. I would like to bring your attention to the middle of the table for citric acid anhydrous. You'll see that RLD product does not use citric acid. On the other hand, test product uses citric acid and listed its function as buffer. If you look at the other components of the test formulation, you'll also notice that citric acid is not accompanied by its conjugate base. Therefore, it does not form a buffer system, and its function is inappropriately listed as buffer. However, citric acid in this case may be more appropriate to call it pH adjuster. Once the function of the citric acid monohydrate is updated as a pH adjuster, it may be acceptable to waive the inactive ingredient requirement if scientifically justified. The possible supporting data here would be supporting information from inactive ingredient database and comparable pH between test and the reference standard products. The key here is that accuracy of excipient function matters. Now let's take a look at some safety concerns when there's a pH adjuster difference between test and reference products. When there is pH adjuster difference between the test and reference products, there may be some safety concerns. One, it's possible that the final product contains novel inactive ingredient that has not been used in an FDA-approved product, the safety of which cannot be established without clinical testing. It is also possible that the inactive ingredient is found in ID, but not for the same route of administration. It is also possible that the concentration of the pH adjuster is above the limit described in the IID, and this includes situations when the excipient amount is Q2 the same as RLD. In justifying for the safety concerns or addressing the safety concerns, please provide supporting information from the IID and or any other information as needed. Keeping in mind that the IID reference should be for the same context of use as RLD product, including the route of administration, dosage form, level and duration of exposure, patient population, and indication for use. The scientific principles around inclusion of buffer in a formulation may support the justification for difference in pH adjuster. Now let's take a look at the content of 31499B waiver request. One thing to keep in mind is that waiver requests must be submitted with the supporting documentation in the ANDA. Under applicable regulations, a waiver request must contain at least one of the following: an explanation why the applicant's compliance with the requirement is unnecessary or cannot be achieved, a description of an alternative submission that satisfies the purpose of the requirement, or other information justifying a waiver. When submitting a 31499B waiver request, the cover letter should include the relevant RLD information as described in the first bullet point, statement describing the Q1 or Q2 difference in pH adjuster for which the applicant is requesting waiver of the applicable regulatory requirement, summary of type of information submitted to support the waiver request, and the location of such information. Now we'll take a look at when to submit the waiver request and how the agency will communicate the decision on the acceptability of the waiver request. The agency recommends that the ANDA applicant submit a controlled correspondence requesting an evaluation of the proposed formulation and the ROD. The applicant may consider submitting a 31499B waiver request to support the pH adjuster difference in the ANDA submission if the response to the control indicates that the proposed formulation does not meet the inactive ingredient requirements applicable to the product, and the applicant believes that this failure to meet such requirements is due to the difference in pH adjusters. If the ANDA applicant chooses to submit the ANDA without utilizing the control process and believes its formulation may not meet the inactive ingredient requirements in 31499A93 or 4 with respect to one or more pH adjusters, we recommend such ANDA applicants submit a 31499B waiver request with the application. A 31499B waiver request and its supporting documentation must be submitted in an ANDA. We will refuse to receive an ANDA for a proposed product intended for parenteral, ophthalmic, or other use that contains a Q1 or Q2 difference in pH adjuster compared to its RLD, but does not include a waiver request and its supporting documentation. An inquiry about a waiver request through control or pre-ANDA meeting request does not constitute a waiver request. The agency will not consider a waiver request unless a request and accompanying documentation are included in an ANDA submission, consistent with the requirements of 31490 and 31499B. The acceptability of the waiver request will be determined during the scientific review of an application, and we will inform the applicant of the decision regarding the waiver request when we act on the ANDA containing the waiver request. We recognize that an ANDA applicant who seeks a waiver for a difference in pH adjuster may also want to seek a biowaiver or utilize an in vitro approach to demonstrate bioequivalence. Now let's take a look at the effect of pH adjuster difference on eligibility to use certain approaches to demonstrate bioequivalence. When an applicant seeks 31499B waiver of inactive ingredient requirements at 31494A93 or 4 for a Q1 or Q2 difference in pH adjuster compared to the RLD, then that ANDA product necessarily does not contain the same inactive ingredients in the same concentration as the RLD for the bioequivalence to be considered self-evident under 322B1. You may recall that certain PSGs recommend that ANDA product be Q1, Q2 the same as its RLD to use a particular approach recommended in the PSG to demonstrate bioequivalence. On the left-hand side, for this drug product, in order to use in vitro option, the PSG recommends that test product be Q1, Q2 the same as the RLD. On the right-hand side, I have included some example of drug products where PSG has such a recommendation to use particular BE approach. And please note that this list is not comprehensive. We note that recommendations in PSG are not binding, and applicants may use an alternative approach if it satisfies the statutory and regulatory requirements. The scientific principles described in this guidance that provide support for a waiver of inactive ingredient requirements under 31494 A93 and 4 for difference in pH adjuster may, in some cases, also provide support for an applicant scientific justification for use of a particular BE approach. It's also important to note the waiver under 31499B is not the same waiver under 21 CFR Part 320. 349.9b waiver request permits an applicant to seek waiver of requirements under 314.92 through 314.99, including the inactive ingredient requirements at 314.94, 8, 9, 3, and 4. The requirements for bioequivalence are governed by the regulations set forth in 21 CFR Part 320. Thus, a waiver of evidence of in vivo bioequivalence is different from a waiver under 314.99b, and a waiver under 314.99b does not also waive the requirements to submit evidence of in vivo bioequivalence. We encourage an applicant who seeks to submit a waiver request under 314.99b for a difference in pH adjuster and use an in vitro approach to demonstrate bioequivalence to contact the agency to discuss the particular approach to establish bioequivalence for that drug product. Let's talk about the mechanism to receive feedback from the agency. You may use a control to request a Q1, Q2 sameness evaluation of the proposed formulation against a reference product. The agency will respond to indicate whether the proposed formulation would or would not likely be refused to receive. You may use the pre and the product development meeting to get feedback on the proposed formulation and the rationale that may support the 314.99b waiver request, where appropriate, for a difference in pH adjuster for a parenteral, ophthalmic, or otic product. That concludes our presentation. On behalf of the presenters, thank you for your attention. Now let's go to Q&A and panel discussion.
Thank you, Dr. Kozak and Dr. Chung, for those excellent presentations. We will now begin the question and answer panel session of the webinar, and it is my honor to introduce our panelists that will be joining Dr. Kozak and Dr. Chung. Dr. Utpal Munshi is the Division Director of the Division of Bioequivalence 1 in the Office of Bioequivalence. He leads a team of scientists responsible for the assessment of the bioequivalence sections of ANDAs and other stakeholder submissions. During his time in DB1, Dr. Munshi has a variety of technical and administrative roles and has participated in the drafting of numerous agency guidances pertaining to bioequivalence. Dr. Munshi received his PhD in biological chemistry from the University of Michigan and then undertook postdoctoral training at the National Cancer Institute in Frederick, Maryland before joining DB1 in 2007. Dr. Melissa Mannion is a regulatory counsel with more than 15 years of experience encompassing public health policy research, regulatory and legislative relations, pharmaceutical purchasing and distribution, professional affairs, and retail pharmacy operations. Melissa earned her bachelor's degrees in human biology and psychology from the University of California, San Diego, and holds a Doctor of Pharmacy degree and JD with a concentration in health law from the University of Maryland, Baltimore. She serves as a policy subject matter expert on a range of complex issues pertaining to generics, including bioequivalence, sample retention, pre-Hatch-Waxman ANDAs, inactive ingredient sameness, disclosure concerns, and importation. Dr. Phung is currently a supervisor in the Division of Filing Review in the Office of Regulatory Operations. He received his Doctor of Pharmacy from Massachusetts College of Pharmacy and Health Sciences in 2010 and commissioned into the U.S. Public Health Service Commission Corps in 2017. Collectively, he has over 10 years of experience in varying capacities. He started with the FDA as a filing reviewer and progressed to project manager and team leader positions. Prior to joining FDA, he has held a spectrum of clinical, administrative, and leadership roles in the U.S. Air Force. Dr. Brittany Avaritt is a policy lead in the Office of Policy for Pharmaceutical Quality in the Office of Pharmaceutical Quality. She received a PhD in Pharmaceutical Sciences from the University of Maryland, Baltimore in 2014. Brittany has experience assessing the bioequivalence of generic drug products and developing and implementing regulatory policies related to the generic drug program and drug product quality. Dr. Bing Kai is Director of the Division of Liquid-Based Drug Products in Office of Lifecycle Drug Products in Office of Pharmaceutical Quality. In his 20-year tenure within the FDA, he has been promoted to CDER Senior Reviewer, Team Lead, Chemistry Division Deputy Director, and Division Director. He has been involved in the development of several important agency initiatives, including the current ANDA Integrated Quality Assessment Process. He has coordinated the implementation of the Comprehensive Review Assessment using quality by design and risk-based review concepts for various drug dosage forms to ensure a uniform drug quality program across generic and new drug products. Let's begin our question and answer session. The first one is for Dr. Phung. How do we inquire about whether a particular pH adjuster difference would be acceptable to waive under 314.99b in a Q1, Q2 controlled correspondence?
Thanks, Amanda, for the question. When an applicant asks FDA to waive a requirement, including under 314.99b for pH adjuster differences, the acceptability of that waiver request becomes a matter of scientific review. FDA would not respond to those inquiries on whether a particular pH adjuster difference would be acceptable to waive under 314.99b in a control. But if the ANDA is submitted with the waiver and the justification for pH adjuster differences in the formulation, and assuming that there are no other issues unrelated to the waiver request that would trigger a refusal to receive, then the ANDA would be received for scientific review. And just as a reminder, if you submit an ANDA for a parenteral ophthalmic or otic drug product with a formulation that has pH adjuster differences without submitting a waiver request and the supporting documentation, the ANDA will be refused to receive.
Thank you, Dr. Phung. This next question is also for you. It is my understanding that for ophthalmic products, FDA has been requiring Q1 and Q2 sameness for all inactive ingredients, including exception excipients. Does this mean that we can request a 314.99b waiver for differences in exception excipients for ophthalmic products?
Yes, thanks again. So for ophthalmic drug products, under the regulation at 314.94A9 at Roman numeral 4, generally requires inactive ingredient sameness, which includes pH adjusters, to the RLD, but it does allow for differences in exception excipients. So for the purposes of the Q1, Q2 assessment of the formulation, an ophthalmic drug product is permitted to have exception excipient differences and would not require submission of a 314.99b waiver.
Thank you, Dr. Phung. So this question is for Dr. Mannion. Along the same lines, it's my understanding that despite a similar allowance to parenteral products provided for ophthalmic drug products in 21 CFR 314.94A9 Roman 4, FDA has determined that as a scientific matter, any qualitative or quantitative deviations from the RLD should be accompanied by an appropriate in vivo bioequivalence study. Can you please clarify how this applies to pH adjuster differences for ophthalmic products?
Yes, for ophthalmic products, so 314.94A94 generally requires the formulation to contain inactive ingredient sameness, right, including the pH adjusters to the RLD, but it does allow for differences in exception excipients. And as we discussed today, a difference in pH adjuster as a non-exception excipient would require submission of a 314.99b waiver request for the inactive ingredient requirement at 314.94. Now, separately, with respect to the approach used to demonstrate bioequivalence, under Section 321 CFR 320.24, applicants are required to demonstrate bioequivalence for their specific drug product using the most accurate, sensitive, and reproducible approach. And while FDA shares our thinking on our approaches that may satisfy this requirement and guidances, which have included recommendations for ophthalmic products to use an in vivo approach where there may be a difference in any inactive ingredient, whether it's pH adjuster or exception excipient. These recommendations are not binding, and applicants may use alternative BE approaches as long as it meets the statutory and regulatory requirements. So for ophthalmic products, the formulation must be both Q1 and Q2 same with respect to all non-exception excipients to satisfy the inactive ingredient requirement at 314.94A94. and must use a BE approach that satisfies the requirement at 320.24a for demonstrating bioequivalence.
Thank you, Dr. Mannion. Dr. Kozak, so if I want to seek a bio-waiver or pursue an in vitro method to demonstrate bioequivalence for my ophthalmic product, but do not meet the criteria for QQ sameness called for in a PSG?
Thanks for that question, Amanda. As you mentioned, so if you were going to propose a formulation that has a difference in pH adjuster and apply this 314.99b waiver, it would generally not consider the QQ the same, and so a BE approach would be considered to be alternative to that posted PSG if it recommends a QQ approach. With that being said, as Dr. Mannion mentioned, the FDA's guidances, including the PSGs, are recommendations and are not binding requirements. So an applicant can use an alternative approach such as this if it satisfies the requirements of the applicable statutes and regulations. To discuss that, we'd really recommend applicants to discuss that alternative approach to that alternative to the PSG recommendation via a pre-ANDA product development meeting. As part of that, the information on a product development meeting or pre-ANDA meeting can be found in FDA's guidance on formal meetings with FDA and applicants. But in brief, what we'd recommend as part of that proposal, you include in your meeting request sufficient background information and rationale for your proposed BE approach, if it's a vitro, bio-waiver, in vivo, if it's an alternative that's requesting this waiver, that will allow us to engage in a meaningful scientific discussion. So this would include the information on the proposed difference in pH adjuster compared to the RLD, as I mentioned in the presentation, as well as discussed in the guidance, as well as how the difference is expected to impact the other ingredients and overall formulation properties. And importantly, how your proposed BE approach can support a BE determination to the RLD. And how you frame a proposed BE approach and the feedback you'll likely receive will be dependent on the information and the potential differences that you're proposing compared to the RLD formulation. I also want to remind folks that although pre-ANDA meetings are generally only granted for complex products, a pre-ANDA meeting may be granted to discuss a proposed BE approach for non-complex products, such as the parental ophthalmic and other products being discussed here, if they're proposing a 314.99b waiver request as part of that for a pH adjuster. Thanks for that question.
Thank you, Dr. Kozak. Our next two questions will be for Dr. Chung. Can we use a pH adjuster different from what is used in the reference listed drug or reference standard and seek waiver for the inactive ingredient sameness requirement?
Thank you for the question. If the applicant's formulation contains a different pH adjuster than what's in the RLD, it would require submission of the waiver request with their ANDA for the inactive ingredient requirements at 314.94a, 9.3, and 4. And as a reminder, the regulation generally requires the parenteral, ophthalmic, and otic drug products to contain the same inactive ingredients in the same concentration as the RLD and not the RS, the reference standard. Thank you.
Thank you, Dr. Chung. One more question for you. Are pH adjuster changes only allowable for hydrochloric acid or sodium hydroxide, or can we use any other pH adjuster?
Yes, thank you for the question. The recommendations in the guidance apply to the inactive ingredients in the ANDA that adjust the pH of a drug product intended for the parenteral ophthalmic or otic use. So, as mentioned during the presentation, we believe that a Q1 difference in pH adjuster in certain circumstances may be acceptable in an ANDA. And keep in mind that the pH adjuster change would result in a will deny scenario as mentioned in the slides, and which are more appropriate for review as a new drug application. If an applicant chooses to use a different pH adjuster or amount of pH adjuster different than what's in the RLD, the onus is on the applicant to provide the supporting information and justification for the request that the FDA waive the inactive ingredient requirement for the proposed difference in pH adjuster. Thanks.
Thank you, Dr. Chung. Our next question is for Dr. Munshi. What sort of studies could be recommended by FDA for complex products while changing a pH adjusting agent?
Thank you for the question. So really, I think this depends on the nature of the change, would be a case-by-case evaluation and dependent on the proposed difference with respect to the RLD. Some examples of that information that would support the difference are already included in the guidance, but specific studies will depend on the formulation and if and how the proposed pH adjuster difference can impact the final formulation. For example, does it alter pH, counterion concentration, introduce a new counterion? These are all aspects that should be considered and explained in the applicant's proposed approach. Of course, as a reminder, the applicants must conduct bioequivalence testing using the most accurate, sensitive, and reproducible approach available among those set forth in 21 CFR 320.24 that is capable of establishing bioequivalence for the product being tested. As a reminder, of course, the FDA regularly communicates our recommendations and our thinking regarding approaches that we believe satisfy this requirement through general guidances, product-specific guidances, and also through pre-submission correspondence and communication. Of course, applicants may choose to submit alternative BE studies, but you would need to provide justification for the use of that alternative approach to demonstrate that it meets the statutory and regulatory requirements for approval.
Thank you, Dr. Munshi. Our next question is for Dr. Kozak. Does the pKa of the drug come into play?
Yes, thanks, Amanda. I think this is a really good question because it's not only the pK of the drug that needs to come into consideration when you're proposing a difference in pH adjuster, but the pKa and the pH of the other ingredients as well as the overall formulation. And that all needs to be considered when you're outlining your rationale and justification for a difference in pH adjuster. An example for that, I think Dr. Munshi kind of mentioned a few, and then the guidance also recommends a few. But aspects there is to understand and kind of explain in your rationale how the proposed difference in pH adjuster interacts with the other ingredients in the formulation. Do they become part of a buffer? Are there interactions with polyonic polymers that are controlling the formulation viscosity, or any other sort of interactions with the other ingredients, such as the active ingredient? So all pK of all components in the formulation are really important to consider and provide rationale when you're proposing your pH adjuster waiver request. Thanks.
Thank you, Darby. Dr. Kai, do you have anything to add to this question?
I think I'm good. Yeah.
Okay, thank you. Well, actually, our next question is for you, Dr. Kai. Some pH adjusters are complex in nature and may influence drug product quality attributes. What are some of the considerations?
Yeah, thank you, Amanda. That's good questions. So, as Darby just mentioned earlier, right? So, a pH adjuster or pH modifier can definitely influence the pH or pH range of the drug product, which can significantly have impact for drug product quality. So I can give you an example, right? For a peptide product, if you use a different pH adjuster or buffer, can have impact on drug product, such as secondary structures, aggregations, even on the stability. So those elements, when you definitely should put the considerations when you want to use a different pH adjuster or pH modifier.
Thank you, Dr. Kai. We will stick with you for the next question. Do we need to consider how a change in pH adjuster might affect degradation of parenteral, ophthalmic, or otic drug products as part of the waiver request?
Can you repeat the question again?
Sure. Do we need to consider how a change in pH adjuster might affect degradation of parenteral ophthalmic or otic drug products as part of a waiver request?
Sure, thank you. Yeah, when an applicant requests a waiver for the inactive ingredient requirement for pH adjuster different in the formulations, the applicant is still required to meet all the regulatory requirement for the approval, right, including the stability study for the product. So as I mentioned in the above cases, right, so different pH, especially when you change to a different pH adjusters or pH different buffer systems, you may have significant impact for formulation-related issues. In other words, will impact to the drug product stability profile. So those all should be put considerations. So I don't know, Upal, you want to add anything to that?
No, thank you, Bing. I think you covered it.
Thank you.
Thank you, Dr. Kai. Our next 3 questions will be for Dr. Mannion. The first question is, can a 314.99b waiver be applied to topical products?
So waivers are for requesting a relief from a regulatory requirement, and topical products are not actually required to have an active ingredient sameness in our regulations. But some of our PSGs call for QQ sameness as criteria for certain recommended BE approaches, such as like an option to use an in vitro approach. And waiver for a difference in pH adjuster would not apply or be needed for a topical product, since QQ sameness criteria and PSGs only reflect the agency's recommendations and are not requirements. And so we would encourage an applicant who wishes to use an in vitro option described in the PSG that includes QQ sameness criteria, but has a formulation that is not QQ same as the RLD, to reach out to the agency to discuss this alternative BE approach to what's recommended in the PSG.
Thank you, Dr. Mannion. The next question for you is, can you please clarify whether an ANDA can add a pH adjuster if none is disclosed in the RLD labeling?
So we understand that there may be limited information available publicly with regards to an RLD's formulation. So we encourage you to submit a control correspondence to seek a Q1, Q2 assessment of your formulation before submitting your ANDA. The controlled correspondence pathway is intended to help applicants with developing their specific drug product by providing feedback on various regulatory requirements, including the inactive ingredient sameness to the RLD. At the same time, we also recognize that due to formulations being trade secret information, FDA's responses to your QQ assessment questions may not always be enough to resolve all of your formulation questions to achieve a QQ same formulation. And so furthermore, if, for example, you have submitted controls and haven't been able to achieve a QQ match to the RLD, and based on the results of your reverse engineering, you believe that a pH adjusting agent is contained in the RLD but not disclosed in the RLD labeling, we would encourage you to submit a control and share that information with us for consideration when assessing your proposed generic formulation for QQ sameness to the RLD. Such information would help ensure the accuracy of product labeling information and support generic drug development. And we are continually striving to improve our review processes and monitoring of FDA-approved products. And your input would help ensure that drugs are safe and effective for use. Nevertheless, if you believe your formulation adds a pH adjuster that is not contained in the RLD, as Darby mentioned in the presentation, an applicant should submit their ANDA with a waiver request for adding a pH adjuster to its formulation, including supportive documentation and justification for the specific pH adjuster difference being proposed based on the information available to you. This will help give clarity to the agency as to the specifics of the request for waiver being made and the scientific justification for why the proposed difference would not impact the safety or efficacy of a drug product.
I have one more question for you at this time. So a number of questions have been submitted regarding applying this guidance to specific products. Are we able to address these inquiries in this webinar?
So actually I would recommend that you submit those specific drug development questions as a control to the agency so that we're better able to address those questions. We are not addressing specific drug development questions for a number of reasons during the webinar. We are here to answer questions that are specific to the guidance being discussed today, and on the general topics around pH adjusters and the associated waivers under 31499B.
Thank you, Dr. Mannion. Our next two questions are for Dr. Avaritt. The first question is, when a pH adjuster has dual functions, for example, pH adjuster plus buffer, whether we treat this inactive ingredient as a buffer, which is an exception excipient, or a pH adjuster, which is a non-exception excipient, is dependent on its main function. Is this understanding of the guidance correct?
Thanks, Amanda. When a pH adjuster functions as both a pH adjuster and as part of the buffer system, we generally apply the function of the inactive ingredient as identified and demonstrated by the applicant. If the inactive ingredient is identified as a pH adjuster, that ingredient is considered a non-exception excipient subject to Q1, Q2 requirements. The 31499B waiver allows for flexibility in the regulatory framework such that a pH adjuster difference, when there is also a buffer contained in the formulation, may be acceptable in an ANDA. Thanks for the question.
Thanks, Dr. Avaritt. One more question for you. Would you please clarify if the pH adjuster was included in the buffer, it will be considered as an exception excipient?
Thanks, Amanda. If an applicant identifies the function of an inactive ingredient as a buffer, then that inactive ingredient will generally be considered an exception excipient. Therefore, it's important for the applicant to correctly identify the function of the inactive ingredient. Thank you.
Thank you, Dr. Avaritt. The next question is for Dr. Kai. For USP ophthalmic generic drug products, is it okay to propose pH range as per USP monograph, or should it be as per RLD? If pH is proposed as per drug product USP monograph, will FDA refuse to receive an application?
Yeah, thank you. Good questions. Although this question may not directly related to today's topic on pH waiver, but this is a very, very common deficiency we have been seeing around generic applications. So again, for a generic product, your target is the R&D product. So even though you have a USP product and you have a USP range specification, that may not necessarily means this is totally. I would say USP requirement is only a minimum requirement. So really, your specification or your target should be aimed to the R&D product. Now, in some cases, the R&D range or spec may not necessarily line up with USP. Again, R&D product is still the golden standard. So if you want to propose something other than USP monograph, those can be review issues when you end approach to the approval or agency. You get a green light from the agency for the pH range, then you always can petition with USP if your range is different from USP specifications. I hope I addressed that questions.
Thank you, Dr. Kai. Dr. Phung, do you want to add anything regarding the refuse to receive aspect?
Oh, yes. Thank you. So as Bing alluded to, the proposed pH range is determined during the scientific review, so that would not be a basis for us to refuse to receive an application. Thank you.
Thank you, Dr. Phung. Our next two questions are for Dr. Kozak. The RLD package inserts often only list that the pH adjuster is used to reach a given pH range. How is an applicant supposed to know if the RLD adds a fixed amount or QS? Should we submit a control correspondence for every potential ANDA?
Thanks, Amanda, for that question. I think, as Dr. Mannion mentioned a bit earlier, we understand that there can be limited information publicly available in regards to RLD formulation, and those formulations being trade secret. FDA's response to these QQ controls may not necessarily answer all the questions you may have about a drug product formulation. With that being said, I think there's a key aspect here is that the control pathway is the most appropriate to ask about formulations assessments from the agency, and reliance upon your reverse engineering and understanding of the drug product. The expectation is you may be able to deduce the fact if a RLD or if this is the aspect that you think may be the reason as to why you're getting a response that your product formulation may not be QQ similar. In those situations, it would be highly recommended that this waiver guidance, the 31499B, would be an appropriate sort of pathway, and you would provide your additional rationale and justification based upon that information of both reverse engineering and the FDA's response to your questions. If you have additional questions or proposals on this could be something that could be discussed through a pre-ANDA meeting. But in general, the QQ sort of questions and response would be most appropriate through a control correspondence. Thank you for that question.
Thank you, Dr. Kozak. One more question for you. In RLDs where pH adjusters are used as QS to achieve an approximate pH, is it recommended that prospective ANDA filers list a range of pH values with a target or approximate pH of X in the controlled correspondence requesting Q1, Q2 evaluation? What is the agency's understanding of the word approximate in this circumstance?
Thanks, Amanda. So one aspect here is when we're doing a QQ assessment and the pH adjuster is being used on a QS basis to a pH, in general, the target pH or the pH that's being directed towards or approximate towards is not a part of a QQ assessment per se. What that is you would not necessarily get a feedback on if that would be QQ similar. It is a part of the overall scientific review when the ANDA comes in, because this would be set ranges with specifications and aspects like that. And saying that, it is always good if you're proposing a pH adjuster with QS to include the pH and pH range there that you're intending to use, but it would not be a part of a QQ response in general from the agency through a control correspondence. Hope that's helpful. Thanks.
Thank you, Dr. Kozak. Our next question is for Dr. Chung. If a pH adjuster is changed to accommodate a base versus acid form of an active ingredient, is a waiver possible if the salt version of the active ingredient is the same as the RLD? How should an applicant pursue listing the ingredients for this API conversion versus pH adjuster waiver for Q1, Q2?
Thank you for the question. So as mentioned during the presentation, what's in the final product should be considered. For example, if you have a counter-ion species that is not present in the RLD, and it can fall into will deny or may deny scenario, and you can refer to the information in the presentation. Another aspect to consider is when you have a pH adjuster that has multiple functions. For example, some is being used to convert the API, while the remaining amount is used for pH adjustment. It's preferable that you clearly list in your formulation table how much is being used for the conversion and how much is being used for the pH adjuster, and that would make our review process much smoother. Thank you.
Thank you, Dr. Chung. One more question for you. How can an ingredient be above the inactive ingredient database limits and Q2 the same as the RLD?
Good question. So it's possible that the Q2 difference between the test and reference product is within the 5%, but at the same time, that difference may be on the high side for the RLD, for example, for the test product. So let's say the amount in the test product is 4% higher than the reference product, then effectively that amount is beyond the RLD, and therefore that level should be justified using the reference from the IID. Thank you.
Thank you, Dr. Chung. Our next question is for Dr. Kozak. In physicochemical characterization, why is osmolality recommended, and can one measure osmolarity instead of osmolality?
Thanks, Amanda. I think this question is in reference to within the guidance, we provided some examples of different types of information, specifically in vitro characterization information, that may be used to support a waiver request. And that being said, what we want you to do is to look at the actual formulation, the pH adjuster difference, and the critical quality attributes, or those types of studies that may be applicable or be different or show sameness of that do that change. So in reference to this question here, the osmolality or even the osmolarity can be used. It would be used if it's a part of the overall critical attribute, or if the change in pH adjuster may be changing that or have an effect on that overall composition. So you want to show that. So I think either could. Those were just simply a list of potential sort of types of information that may be supplied. But what we want to make sure is that applicants are looking at their actual proposed formulation at the RLD, understanding what difference could actually happen, and theoretically show why these tests or this additional information supports that it would be no different to that reference listed drug product. So thank you for that question.
Thank you, Dr. Kozak. Dr. Kai, do you have anything to add to this question?
No, I think Darby mentioned it very clearly. I just want to add, these two tests may be used interchangeably. Most time, I don't know if this is for balsamic, when the solution is very dilute, these two values are almost all the same. The reason we use one term versus the other term maybe depends on your method or based on your calculation. But like Darby mentioned, as long as you have adequate justification demonstrate your test product and the reference product are same, then you should be okay.
Thank you, Dr. Kai. While I have you, I have another question for you. Many times the label of the reference listed drug or reference standard product for ophthalmic list a pH range, not a single pH value. Can a generic product have a pH value within the range specified in the label but not exactly matching the RLD or RS pH value?
Sure, thank you. Good questions. Yeah, again, we have been seeing this kind of situations a lot when we see those generic ophthalmic products. So again, as I mentioned, right, the R&D product is always the golden standard. So if they put the label, their range, and you should really formulate at the range according to the reference product. But then we also encourage you to test the reference product, right? So over the year, things may change. So in some cases, their formulation may be changed over the year. However, the label never changed. So if you do see a mismatch during your development, we do encourage you to submit a control to ask an agency for clarifications. This can be a very good questions. So you never know what can happen, right? So on one side you have R&D labeling, the other side you have their product, marketing product. So if they be not consistent, so in those cases, we do recommend that you contact the agency for clarifications. We certainly don't want you make any mistake and then put your product, submit your application, then later find that there was a mistake. But yeah, that's a good question. Thank you.
Thank you, Dr. Kai. Our next two questions are for Dr. Mannion. The first one is, if a product has a pH adjuster listed on their carton and it is established that it actually not required in the manufacturing process, do we need to send a controlled correspondence to request a waiver that it is not used in a suspension dosage form?
So the regulations require that your proposed product contain the same inactive ingredients and the same concentration as the reference listed drug. So, particularly if the, ingredient is you see it on the labeling as being part of what's, in that drug product, and it's not an exception excipient, that would be subject to the inactive ingredient requirements. You can send a control to ask for, a Q1, Q2 assessment of your formulation against the RLD to help maybe address some of this question. But in order for you to meet the regulatory requirement, the inactive ingredients and the concentrations thereof would need to be the same as what's in the RLD. Thanks for the question.
Thank you. One more question for you. For clarification, can the waiver be submitted before ANDA submission in controlled correspondence or only in the ANDA submission?
So 314.99B waivers per our regulations require that you comply with the requirements at section 21 CFR 314.90, and that actually does require that such waiver requests be submitted in an application or an amendment or a supplement to an application. Inquiries submitted via controlled correspondence or asked, for example, in a pre-development meeting would not constitute a waiver request. And so if you would like the agency to consider a request for waiver, that does need to be submitted as part of your ANDA. Thanks.
Thank you, Dr. Mannion. Dr. Chung, this next question is for you. Many reference listed drug reference standard product labels include a pH range, not a single pH value. Can the generic product be within the pH range and not exactly match the RLD-RS pH value?
Thank you for the question. I think, we'll have to take a look at the product-specific scenario in determining the acceptability of the pH difference. That said, we do look at the final pH of the test product as well as a reference product, and also consider the pH specifications and see how they play. So we would like you to keep that in mind when you develop your generic product. Thanks.
Thank you, Dr. Chung. One more question for you. After the agency approves a Q1, Q2 composition via a controlled correspondence, can it be assumed that the pH adjuster given there by sponsor is also accepted by the agency?
So this is an interesting question. So I think some of the things that comes to my mind is that it's possible the RLD labeling is updated between when the control is reviewed versus when ANDA is ready for technical review. So there are some technical considerations, and during the control review, we may be looking at the formulation just at the face value, whereas when we do the technical review, there may be additional considerations. Yeah, there may be a situation where the conclusion of the control may not always carry over to the ANDA review, but in general, it should be okay. But I would add some disclaimer to that. Thanks.
Thank you, Dr. Chung. Our next question, and possibly our last question, is for Dr. Phung. How long does it take to get an FDA response to a controlled correspondence for Q1, Q2 sameness evaluation? And when is the best time that an applicant should request a pre-ANDA development meeting? Which is better, controlled correspondence or pre-ANDA meeting?
Yeah, thanks, Amanda. That's a good question. There are definitely different timelines for control correspondences, whether a drug product is complex or non-complex drug product. And of course, pre-ANDAs also have a different timeline. But we suggest that if there's any discussion that the applicant wishes to have with the FDA, that the best course would be a pre-ANDA meeting to discuss any scientific or regulatory concerns with the proposed formulation. Thank you.
Thank you, Dr. Phung. Dr. Kozak, do you have anything to add to this question?
Yeah, I think Dr. Phung definitely covered the key aspect, but I did want to kind of highlight for getting a standard sort of just a response to if your formulation is QQ the same, the control correspondence route is still probably the most appropriate route for that. I think the aspect here is when you are going to propose a difference in pH adjuster, you've built a scientific rationale and justification, and you're going to propose what type of information may be included. You see that this may be different to a posted PSG QQ approach. And so what you want to do is have that discussion with the agency on the appropriateness of that approach and the overall sort of information. That would come in through the pre-ANDA meeting process. And as I mentioned in earlier responses, what's critical to that is that you provide us with sufficient information and rationale in that meeting request so we can have a good scientific discussion, and that will also let us to be able to grant that sort of meeting. If there's insufficient information, you may receive a deny to the meeting request and additional information that may be needed. In general, my understanding is that controlled correspondence for a standard QQ control is about 120 days. For a pre-ANDA meeting, you'll get a response on the grant or deny of the meeting within 14 days, and then the meeting will be held either through virtual, face-to-face, or written response within 120 days after that grant deny time frame. Thank you.
Thank you, Dr. Kozak, for answering that question. And a huge thank you to our speakers and panelists for sharing their knowledge and expertise to help us understand this new draft guidance and bring to light some considerations as we begin to implement it. We hope you all found this useful, and thank you so much for joining us. And now I turn it over to Dr. Lim to close out the webinar.
All right. Thank you so much, Dr. Jones. And again, thank you to all our presenters and panelists for a very informative session. Just a few housekeeping reminders to close. It will, again, just take us a bit of time to process, but soon you'll be able to access today's recording from SBIA's webpage. Again, please note that the evaluation and certificate are only available for 2 weeks, so the deadline date is August 24th. And another reminder to visit our Cedar Learn webpage for SBIA tutorials and other educational resources. This concludes today's webinar. We hope to see you again at our next event. Thank you.